It's a great honor for me to introduce Professor Shivashankar for our next talk. Prof. Shiva's research investigates the role of cell mechanics and nuclear mechanics in chromatin organization and transcription regulations. His lab's research identified novel approach to induce cellular rejuvenation and reprogramming. In recent several years, his lab developed a chromatin fingerprint-based approach for the disease diagnostic and drug discovery. Shiva received his PhD from Rockefeller University, and after that, he became faculty in National Center of Biological Science. And in 2011, he joined National University of Singapore and co-direct Mechanobiology Institute. In 2021, he joined ETH Zurich and lead a laboratory of multi-scale biology there. Let's welcome our next speaker, Professor Shiva.
Great, I hope. So let me thank the organizers for having me here. Also, it's always a pleasure to have your former PhD student introduce you. Lucien was a former PhD student. Yeah. Good. With that, since it's a very broad audience, what I'm going to do is give you a flavor for the kind of questions we ask and how we approach them. And most of the work that we do is in joint collaboration with Caroline Wooler's lab here. So it's a wonderful morning session, so I don't have to go introduce too much. Many things that many of you have already talked about. Nevertheless, so as all of us know, I don't have to belabor this, we have about 36 trillions of cells in our body. It's the same DNA, so it has to packed in different tissues in a tissue-specific manner. Also, as all of you know, different tissues have different stiffness. Our bone tissues are very stiff, whereas brain tissues are very soft. So one has to try and understand genome regulation in the context of the stiffness. So what I'm going to do in the first half of the talk is to give you some basic principles that we've discovered of how cells operate. We'll then go forward in how we can embrace AI to learn about cell states and cell state transitions. One of the things that we discovered, we and others, of course, identified was that chromosomes, while you pack the DNA into the nucleus, the interfaces between chromosomes have been not well appreciated, right? The intermingling regions between the chromosomes, which can have trans interactions, seems to have a bearing on how transcriptional control come about in a cell type specific manner or tissue type specific manner. Once you think of chromosome interfaces, mechanics becomes important because it's like a box in which you pack chromosomes. If I change the nature of the box in terms of the mechanics, then you have altered genome regulation, which is what I'll show you in the first part of the talk. So over the years, we and others have laid out some of the principles that how cells send signals from the extracellular environment, because cells send both chemical as well as mechanical signals. And these cells then integrate into different parts of the genome when you activate specific transcription factors. So these transcription factors then bring about groups of genes which are co-localized for co-regulation. And so this kind of an idea already suggests that there are really two spaces that we need to worry about. The first space is really an architectural space, which you can probe by imaging. Jennifer talked about quite a bit of the imaging modalities. And of course, this physical space intimately linked with the biochemical space. The biochemical space of cells are highly multidimensional, lots of biochemical networks. One wants to understand causal relationship between this physical and biochemical space. If such a thing exists, we should be able to test it in very simple models, right? So one of the things that we tried out early on was take a cell. These are general principles. You can work with any cells of your choice. Take a cell, change its mechanics or change its geometry, as you see here. There are many methods in cell biology where you can have micropattern surfaces where you can change the shape of a cell. So when you change the shape, it's the same cell, right? When you change the shape of the cell and take its transcriptome, the transcriptome is very different, right? All I've done is taken a cell, changed its shape, right? And the DNA is packed, but what it produces is very different proteins. Somehow the cells adapt to what they see, trigger biochemical circuitry in the signaling pathways, and trigger remodeling of the chromosome interfaces because I'm changing the shape of the cell and the shape of the nucleus, and I produce different proteins. Right? This is very generic of all cell types that you work with. Right? The second important principle is if you now stimulate these cells with some signals, it could be mechanical, it could be chemical signals, the cell state, the cell mechanical state dictates what the output is. Right? So here is an example that I take the same cells, put them in a tissue, organoid, or an engineered tissue, or look at the same cells in tissues. What you find is that every cell has its own local state, in culture or in tissues. And now when you stimulate these cells, every cell has its own program. So that's why fingerprinting every cell's transcriptome in a tissue is so important to understand the biology of how cells work, how cells transition to different states, how cells transition to disease states. Right? In the same cells, same signals, either mechanical or chemical or both, but they turn on very different expression patterns, protein production. So what this led us to believe is that there must be cell autonomous properties, that cells could actually switch states. Here they're not still switching their state. Maybe cells could actually switch their cell states. And so we came with a very simple hypothesis. If a cell has to maintain a memory, because all of us, as we know, we don't have billions of cell types, we have about few hundreds or few thousands of cell types depending on who you talk to. Now, cells have to maintain memory, right? When the cells maintain memory, they maintain memory through their transcriptional program because they produce the same proteins after division. But they also have to maintain this mechanical, geometric memory, right? And that's critical. As I said, the physical space and the biochemical space are intimately linked. So they really need to remember their mechanical constraint as well. So when you look at cells in culture or in tissues, when they divide, they remember the mechanical memory, but then they also remember the transcription memory because they're interlinked, right? So we came with a simple hypothesis that let's break one of this memory element, maybe break the mechanical memory of a cell. As they divide, maybe then they should really forget their transcriptional memory. So we impose this confinement so that cells cannot stick outside. And when the cell divides, one cell was sitting in a good real estate piece, and now the cell divides 2 cells, 4 cells, then there is no space to sit. Whereas if the cells divide in a tissue or a culture, they remember both the memories. And here, as they divide, they start piling up on themselves because the mechanical state is changing. One cell was having a tight geometric constraint. When the cell divided, 2 cells, 4 cells, they're losing their geometrical constraints. Right? And we wondered that this probably could induce alterations in chromatin states, signaling, transcriptional programs, and we had a lot of luck with this simple idea. They're starting from one cell. This is again true for, it's a very generic property of cells, that imposing geometric constraints and dividing them, what these cells ended up doing is you start with some cell type, this is a mesenchymal fibroblast. As they divided, they switched their cell state from a mesenchymal to epithelial. They continue to divide, and then they become stem-like. All I've done is there is no exogenous factors. I've taken a cell, I've worked around the phase space for cells with a simple hypothesis that if you break the mechanical symmetry or geometric symmetry, these cells end up as a stem-like state, right? This is with very high efficiency. That means that this is a very default natural cell state transition that our cells have in our tissues. Of course, they have to maintain homeostasis because you don't want to have diseases all the time. If cells switch their states, we're all unstable, right? Cells have to have some stable solutions. But loss of this mechanogenomic homeostasis then can actually induce partial reprogramming. Now we have shown that we can actually take 8 cells, take them into more younger cells by rejuvenation, and I'm not going to talk about that. But this idea suggests that this could be a precursor for generating a disease state, because diseases start from single cells, right? So how do we understand then, at a cellular level, how do diseases start? And our claim is that maybe this kind of a mechanochemical constraint the cells have in our tissues, with alterations in the mechanogenomic homeostasis, could have this propensity for a cell state switch, right? So we are now searching for such cell state switches in real tissues. I'll talk about one example. And as you know, these are early drawings from Cajal almost 100 years ago, where he noticed with simple microscopes that cells do switch in the microenvironment without any exogenous factors. That means that the microenvironment has adequate information for cell state transitions. So it's been long appreciated that in diseases like aging-related diseases like cancer or neurodegeneration, you don't switch overnight. Cell state transitions happen over a long period of time. So this is, for example, in different types of cancers, as a function of aging, different years that it takes for a single cell in a stromal environment, in a tissue, switches its state and start, abnormal loss of homeostasis leads to growth of a disease, right, over a long period of time. So this is what we want to try and understand. So one of the things the main thing that our group does is to use the lens of how the DNA is packed in each of these cells and see how alterations in this genome could lead to these cell state transitions. So as I said, as we age, as cells age in our tissues, there are very subtle alterations in the chromatin state, how genomes are packed. And so we are very keen on reverting it back or use these changes as biomarkers for health and disease. What I'm going to talk today more is a little more of our efforts to think about the way DNA is packed as an exquisite biomarker for cells' health or disease. Right? So towards that, together with Caroline's group, over the years, I'm not going to go into all the details, over the years we've developed a number of machine learning models to learn how DNA is packed, how the hierarchical nature of folding is set up in living cells by staining various markers of DNA-associated proteins, modifications in the histone that pack the DNA, and various other regulatory molecules that are within the nucleus. Through this set of learning and either supervised or unsupervised learning methods, we have developed what are called chromatin fingerprints for cells' state, cell health or disease. And these fingerprints are large. It's like taking a cell, looking at how the DNA is packed, in the same idea as taking a cell and taking a single cell transcriptome. A single cell transcriptome will give you a whole bunch of gene expression changes that are associated with the cell. Likewise, what we have is a high-dimensional information of how the DNA is packed, because as I said, these two spaces are intimately linked with each other. Right? And so through this, we can now look at cell states very easily now. Right? So one example that we tried early on was take cells of different cancer states. These are cell lines derived from various stages of breast tumor progression, and look at how the DNA is packed in these images. And what you already see is that you have distinct states of chromatin in these cell types. Right? you can actually see that there are very distinct types. So each dot is a nuclei, is a high-dimensional information of the DNA packing, and now you construct a low-dimensional map of how the chromatin structure is set up. And now through a number of methods that I'm not going to go into the details, because diseases don't happen overnight, or cell state transitions don't happen overnight. They happen over a long period of time. It's so hard to record, image the cell state transition real time in a tissue context for a disease progression. So we have developed methods where if you had pieces of information during the progression of a disease, what we can then construct is transport models, take these information to a latent space and transport the distributions of how chromatin state is set up in one cell state and how they change over time. And if you did that, you can then develop the envelope of how the chromatin state transitions change in a disease context. So these kind of things can be very useful when you really look at complex diseases. Here is an example of breast tumor progression. This is a ductal carcinoma that becomes invasive carcinoma. These are human breast tissues, tissue microarrays, and we image the entire tissue microarray of different stages of the disease and fingerprint every nuclei for its state of chromatin. So we've developed a scoring system that we can then annotate a score for this nuclei, whether health or disease state, and the degree of disease state. So using such a scoring system, as the disease sets in, what you can see is the cell state transition that happened through the lens of how the chromatin is changing in this context, right? From a ductal carcinoma to invasive carcinoma. So you get high dimensional readout of the chromatin packing states and how these packing states can teach us about the biology of cell state transitions now, right? And using such methods in collaboration with Caroline's lab, and in particular Shinny, she'll talk about some of this, I think, in her talk. What we developed is a way of taking all the cell state that exists in these tissues and cluster them from healthy to disease states. And what you see is that this kind of a clustering method can annotate fraction of cells that are in the healthy state, how the fractions of these cells change from the health state to the disease state as the disease progresses. And one of the surprising things, to me at least, that came out of this effort was that if you fingerprinted the chromatin states of the cells in different disease states, different stages of the breast tumor, you start seeing that even in the healthy tissue, there are residual cells which have the chromatin fingerprints of a disease state. Right? Although they're not disease state, they're annotated by a pathologist as normal, but there are residual cells. And it seems then that as the disease progresses, you have spatiotemporal reorganization of these fraction of cells that make these ducts into very highly metastatic duct. So this allows us to trace cell state transitions through the process of how breast tumor progression happens. In fact, there's a big unmet medical challenge even today around the world that we cannot predict which tumor becomes aggressive and which tumor doesn't become aggressive. There's an over-treatment. As soon as an individual is detected of ductal carcinoma, only 30% of them go on to become invasive ductal carcinoma, but we cannot predict which one will become invasive and which one will not become invasive. So there's an ongoing project in the lab to try and figure that out now. Can we really figure out through the lens of chromatin states which states are aggressive and which states are non-aggressive? So with this, what it suggests is that if imaging modality can give us so much about the biochemistry, there must be really good ways to translate information from one domain of the data to the other domain of the data. Again, here, I'm not going to go into all the details. We wondered about this because I think these are some of the work done in Caroline's group was actually initiated this multi-domain data translation methods in the field early on. And what you see is a picture of cells and the nuclei and the DNA. Because the DNA is folded in a very hierarchical manner, this should be able to tell us, maybe to predict what could be the transcriptome of this cell, or what could be the genomic architecture of this cell, that are very hard to measure, right? These types of data-intensive measurements in terms of omics technologies are pretty expensive if you want to do it on a whole body scale, whole tissue scale. But what is a cheap data modality is, in imaging, is a cheap data modality. I mean, depending on who you talk to. If you talk to Jennifer, she uses very high-end machines. That might be little expensive. But still, it's much cheaper than taking omics data of cells. So through this number of iterations, what we are now shown is by taking pictures, single-cell pictures, and learning how the single-cell sequencing could be in those cells, even with unpaired data, we can now start annotating what could be the transcriptome of cells from an imaging data modality. Also, these are challenging multi-domain data translation because imaging pixels, whereas sequencing is numbers. So there are many methods now that allow you to go from this space to various spaces. And now in recent work, we have also shown that not only you can go to transcriptome or the ATAC-seq or the high-C of that cell from an image, but we can also go to more biochemical circuitry of the cell, because ultimately you want to understand the regulatory modules. So we can convert that into gene regulatory networks that's going to come out, or Shini will talk about how you can predict protein localization from imaging space, where do the proteins go and sit, and so on and so on. And this is a very powerful enterprise now around the world, that because a cell is complex, has many, many different dimensions of how the data is represented in a cell. And if you want to understand the cell, you need to be able to annotate most of these different data domains, but it's hard to acquire all the data domains of a single cell. So these kind of translation methods, data integration methods, would be valuable, particularly if you can take a simple picture. So we got excited about this because if there is so much information in a picture, we should be able to enable early diagnostics, because it's so hard to know where the diseases start in the body. But maybe if we come up with a simple hypothesis, we might be able to succeed in early diagnostics. So that's what we did. It's based on an idea that, as all of you know, any diseases that start in the body secrete signals. Whether it's tumor, whether it's neurodegeneration, whether it's metabolic disorders, they all secrete signals to the local environment. And these signals, which are chemical signals, end up in the blood eventually. And our blood cells, the immune system, is the most sensitive system in a body to detect any subtle changes in the secretome signals in the body. So our hypothesis was that let's image the blood cell chromatin state, the peripheral blood cell, white blood cell chromatin state, and ask, could that be a detector of the physiology? Right? So if we can actually demonstrate this hypothesis, what that tells you is that not only it tells you about immune function, we can annotate that, but it also tells you about early diagnostic approaches. So here is an example of taking from a tumor, healthy donors and a tumor patients. I think this was head and neck cancer. We take the peripheral blood cells, collect the blood from the body. It's very easy to collect. Take pictures of the blood cell chromatin state. Right? And through these, we could already show that with very high precision, very high sensitivity, we can actually distinguish between cancer patient cohort and healthy cohort. Right? So that means that because the immune cells are so sensitive, we could actually detect very early on signals that are starting from the disease. Right? So this has led into 2 successful finishing trials now, clinical trials in Switzerland, where we can actually now ask, can we then use this not only for early diagnostics, but efficacy of treatments? Because there's hardly any good method in the world today, once you're treated for a disease, about the relapse. because the imaging method that we have has a resolution of 1 millimeter when you take different imaging modalities. But as you know, diseases start from cells. If you delete the disease, you want to ask how much have you deleted. So this is an effort to look at proton therapy for advanced tumors. This is the trial is finished, and actually we're now putting out the data on the paper together with Caroline and the student Hannah. And what this trial suggested to us is that it's a very systematic trial. You collect the blood before therapy, during therapy, after therapy, and post-therapy to see how the immune cell chromatin state is changing. Is it coming towards the healthy cohort, or is it staying as the disease cohort? Right? So we can actually now, with this method, annotate every patient level trajectories of how the disease progression and disease therapeutic efficacy can be annotated. Likewise, we have another trial that got finished. Can we detect metabolic disorders using this chromatin states of blood cells? Because metabolic disorders are a big disorder in the field. And is it possible to use cost-effective methods as this, simple images, to give you insights on metabolic disorders? Again, one of a colleague who did this is in the audience somewhere in the lab. So this has motivated us to move towards a large public health project where we would seek all of your help, both in terms of funding, but also participation in the public health project. What we are now trying to do is, can we then fingerprint blood cell chromatin states of individuals? If we have a map of that, of healthy individuals, then that's a reference map that we can create. So this is what I'll end with, that we have started this project in partnership with the Swiss Red Cross. Because how do you define who is healthy and who is not healthy? Because every clinician will not agree to their own interpretation. So what we decided to do was to partner with Swiss Red Cross, where they have a series of checklists to make sure that a person who donates blood is healthy with some boundary conditions, and this blood is implanted in the other individuals. So we're having that checklist as a measure of healthy individuals. And so we are now taking blood from a large cohort of Swiss citizens across different age groups and male and female. The idea is to create like a chromatin imaging atlas of blood cells, peripheral white blood cells, which is so informative of the physiology in the body, as a map that will give us ability to do early diagnostics. Because then any outlier of the chromatin state outside this would be an annotation for a disease. So we are now large clinical trials starting with one of the biggest hospitals in Switzerland to really fingerprint patients who come for early diagnostics through PET scanning, to collect the blood before the PET scan and look at the blood chromatin state and fingerprint, and we have all the metadata of the patients, to fingerprint how their chromatin states of blood cells look like in the context of various diseases, and use the healthy chromatin state as a reference map, right? Because you need a good reference benchmark to say, and the heterogeneity within, because every one of us is very different. Even though we are healthy, if I look at the blood from each one of you, the chromatin state is very different depending on what you've eaten, what is your metabolic activity, whether you have small cough, cold, so on and so forth. So we need to map that envelope of the heterogeneity, which is a fingerprint, which is an atlas that we need to create. And this is a large project, and then we're also taking it towards precision or simple optical methods that can be that these blood cell chromatin states could be visualized on a small microchip that's connected to a very highly sensitive camera, the microscope that we have built, and we're now prototyping the second version, that can connect to a simple mobile phone, so that then we can actually take it to scale up the process, because then we can actually fingerprint a lot more volunteers around the globe. So right now this is a Swiss-based program, and we'll eventually make it into a larger scale program. And we're of course writing to a lot of foundations to fund this. With that, let me end. So what I took you through today is very simple ideas of a cell that sits in a tissue microenvironment, senses both mechanical and physical signals, mechanical and biochemical signals. We're used to thinking of biochemistry more, but it's important to realize that mechanochemistry is what cells see in our tissues, right? And that signals integrate to cells, and that with the loss of homeostasis, you can induce a cell state transition. So what I showed you today is how general principles exist in our cells to integrate mechanochemistry, transduce them through canonical signaling pathways, remodel the way chromatin structure is set up in these cells depending on the microenvironmental cues, and that gave you outputs, differential outputs for the same cell, same signals, but very different outputs because of the mechanics, right? And based on this, what I showed you also is that we can induce cell state transition, because that could be the precursor for understanding cells, how they switch into diseases in tissues. And that's what we're now looking at with lots of this tissue-based analysis that we're doing. And on the other side, what I showed you is the way that our genomes are packed and its intimate link with biochemistry, complex multidimensional biochemical networks. The images of chromatin gives you a fingerprint for that cell state. And that fingerprint is very resourceful, valuable, because then we can say the health of the cell or disease in any complex environment, right? Whether it's neurodegenerative tissues, early developmental programs, differentiation programs, or disease programs, we can then fingerprint cell state transitions with that. So with that, let me end. I think my time is up. Let me thank a wonderful set of colleagues who come from very different areas: computer science, cell biology, molecular biology, and engineering and physics, and a number of scientists that are part of this program. And this work that we do is in close collaboration with Caroline's lab here at the Broad, and a number of very talented colleagues in our lab contribute to our ongoing programs as we move forward. And I think Shinny, one of the very talented colleagues of us, she will present in this session, I think. Yeah. Okay. With that, thank you so much.
So I find the idea of actually trying to predict things from blood very powerful, because it's very non-invasive and it's sort of grail. And I can see how metabolic diseases have a very strong signature, because the high metabolic state highly impacts chromatin regulation. But a lot of diseases result in like this massive inflammatory signal that sometimes is, at least I play with gene expression and single cell expression. It's hard to sort of dissect the different inflammatory diseases. The inflammation signal is just so overwhelming. So I'm wondering if you begin to see differences, or how you would dissect the different onslaught of causes of inflammatory signals in your approach.
Yeah, no, that's a great question. So one of the things that we wanted to first identify is, can we distinguish between a cancer versus non-cancer, another disease, right? So the reason I say it is that you get a disease-specific secretome in the blood, right? And so the chromatin states alter depending on the signals, right? So if it is a cancer signal, the chromatin state remodeled in one particular way, versus if it's a neurodegenerative signal that we are now looking for, chromatin reorganized in a completely different way in a metabolic disorder. Of course, the output is single cell transcriptome that we are used to measuring in immune cells. The idea that images of chromatin has a powerful fingerprint in the way that chromatin can alter, because you can't alter chromatin in many different ways. because we only have a finite type of cell states in the body. So there are only allowed configurations that chromatin can switch. This is what we are now testing. That's why this large-scale project is to fingerprint what are the allowed states of chromatin that it can go to. How can we distinguish between just inflammatory response versus disease-specific responses, right? And this is an ongoing program. Any ideas, happy to get from you. But what we can already distinguish is that given that different secretome gives you different chromatin states, and it's easy for us to scale up, we can then get very good statistical insights on the chromatin state, allowed chromatin states in different contexts, right? How early we can detect, I don't know at the moment, right? These are late stage detection that we have shown, but we are hopeful that we should be, if we get this, healthy donor atlas as a reference map, we should be able to detect outliers much more easily. Because the healthy donor atlas will tell us about the variability that can exist in the chromatin state of blood cells depending on all the individuals, right? Even 10,000, we are now trying to do for 10,000 donors. I don't think that's enough. We need to scale that up more. So we are in the process of, yeah.
One quick question.
Yeah.
Just very practical, there may be others competitive, biomarker, for the early detections of the breast cancer, and I just crossed the paths on the, clinical research group, and my quick question is that, did you find the positive predictive value of your studies, and how does it compare to other biomarker, whether I should pick your, chromatin structure as an initiative for public health or some others?
Yeah, that's a fair enough question. The space is too large, right? Because early diagnosis is a big field. It's billions of dollars because it's so important. There are very competing technologies. The most mature technology is this ctDNA. Disease cells secrete DNA pieces into the blood, and the modifications of the DNA pieces in the blood is a powerful biomarker for disease, right? There are other competing technologies, exosomes. Disease cells release small vesicles and proteins inside them. There's a lot of mass spec proteomic approaches for detecting exosomes in the blood. And circulating disease cells, circulating tumor cells, circulating disease cells, the other technologies that. What we are suggesting is a completely orthogonal approach, because by the biology of the immune system, how sensitive they are, we think that the chromatin alterations could be yet another modality, and a cheap modality, because in the field, if you want to detect early, I can't take a sequencing machine or a mass spectrometry machine, but I can take my mobile phone if we succeed. So now the benchmarking is what we are doing with the atlas that we are doing. Give us a year or two, and I'll tell you how it competes with other technologies. Yeah.
Thank you.
Yeah, thank you, Shiva, for this wonderful talk. I will hand over to Dora.